And so in sum, studies like these have allowed us to put together a complete timetable that is rigorously adhered to for organellar replication in these parasites, beginning with the replication of the centrioles, packaging and successive packaging of the Golgi apparatus, the plastid, the nucleus, assembly of this daughter scaffolding, which then picks up the endoplasmic reticulum and the mitochondrion, and finally the specialized secretory organelles, the rope trees and micronemes, are assembled de novo in each parasite. So in answer to the question that we started with of how do we build a parasite, we do so with a process that's significantly different than the familiar cell cycle processes that have been defined in yeast cells and in mammalian cells, where the hallmark marker of S phase, DNA replication, is completely subsumed within the process that we normally think of, processes we normally think of as associated with M phase, organellar replication, mitosis, cytokinesis, this large region indicated here in pink and encompassing approximately 80% of the parasite cell cycle, including the DNA process of DNA replication. This argues certainly that there are likely to be significant modifications on the familiar checkpoints associated with cell cycle control and changes that will be interesting to explore as we characterize the biology of these parasites further. So in answer to the question of how we build a parasite cell, the answer appears to be that we hang everything onto the cytoskeleton, building the parasite from the top down in a process that ensures the maintenance of polarity that's so essential to parasite survival, and has a number of other interesting implications as well, because everything in the daughter parasite is put there by choice. Residual material, waste products, for example, are sloughed off behind, allowing the parasites to survive without classical lysosomes. So I hope that this tour through the biology of Toxoplasma parasites and the ways in which we can use this as a model organism to study the biology of apicomplexan parasites in general has given you some insight into the fascinating cell biological processes that are involved, both parasite-specific features and features that are more general to eukaryotes. And I hope it will encourage you to read more about the biology of these organisms, perhaps to work on these organisms yourself.